All right. Good morning, everybody. Today is Friday, April 27th, 2018. And this is Before the First Cup of Jules here on UCY.TV. Happy Friday, everyone. It is a cool 47 degrees and overcast, but soon to be springy again here in upstate New York. So where do we leave off on Wednesday? We titled the show Levi's DNA, that was the spit in the cup, Earth Biogenome, and that was the Noah's Ark of genomes of every living creature on the planet. Blockchain ID, iMotif, and that was the weirdly newly found shape of DNA, and biowarfare. And we looked at Fort Detrick, Maryland, back in 1977, I believe, where they were already starting to develop genetic research projects. And after they cleared out all the biowarfare people, they just kind of took up a new game. And now, of course, we are worried about the democratization of the ability to utilize CRISPR to snip, zip, and unzip, mix and match DNA to create who knows what in mommy's back bedroom and in rental units in some garage somewhere. It's just very frightening. So I think that's where we are going to continue today. There was that iMotif, the title of the story that we read out of Popular Science was, scientists just discovered a strange new DNA shape lurking in human cells, and it's all knotted up and crazy stuff. But yes, March 9th, 1977, genetic research at Fort Detrick raises few hackles in Frederick, Maryland. Well, I did a little bit more searching after the show on Wednesday, and I came up with something out of NPR, npr.org. Fort Detrick, from biowarfare to biodefense. Now, if you remember, we had talked about that document that banned bio and chemical warfare that Nixon inked back at the end of, the document was ratified in the 60s, and he inked it in 1972, ending our biological and chemical warfare development efforts, wink. All right, so the story, August 1st, 2008, out of NPR. Fort Detrick, Maryland, was created in the middle of the World War II and became the center for the America's biological warfare efforts. But that role shifted in 1969, the government says. Yeah, the government says, to focus solely on defense against the threat of biological weapons. Now, very interestingly, I'm reading this book I mentioned that Camille suggested to me. Is very excellent. It's called The Biology of Doom, I believe, by Ed Regis, and he goes through the history of the biological warfare program. And do you know who the very first person was that they appointed to run that program? Well, you could have guessed it, but Mr. Merck, who started then Merck, the vaccine and drug company. So I thought that was pretty interesting. And of course, it was all in a defensive posture that these technologies rolled out. And then remember, we talked before about how even now you get military funding in all these universities and companies and laboratories, and then they come up with some new technology and they spin it out with an initial public offering and create all these new technology companies that they have their slippery funding tentacles into. So back in the 1969, the government says they shifted their role to focus solely on defense against the threat of biological weapons as opposed to developing them. Then called the Detrick Air Force Base, the science and research facility housed 4 biological agent production plants. What? Anthrax, of course, was considered the most important agent, and simulants were tested, and 1 bomb was ready for production in 1944, and that's the whole beginning of that book about the biology of doom. That's what it's called. They talk all about developing biological bombs. 1 million bombs were ordered though the order was canceled when the war ended in 1945. Well, thank God. And during the 1950s, the biological weapons program was among the most classified within the Pentagon. There was an emphasis on biological agents for use against enemy forces, as well as plants and animals. And that's where that Dr. Huber was involved, studying the research that the Russians had been doing. The Army said that no biological weapons were used during the Korean War, wink, though such allegations were made by the Chinese and the Koreans. One plan at Fort Detrick in the late 50s was to use a yellow fever virus against an enemy by releasing infected mosquitoes. Oh, go figure. Now, we've talked about some of their tinkering with mosquitoes, and the Japanese did some research in using mosquitoes as flying vaccinators. And there was research in the U.S. that was being done utilizing what they called entomological vectors, and that would be using insects to spread disease such as plague and things of that nature. Detrick's lab was capable of producing 500,000 mosquitoes per month, with plans for up to 130 million per month. The military also tested bombs in Utah, and I read about this one in a book, with Brucella suis, a bacterium that can lead to fever and an influenza-like sickness. The scientists at Fort Detrick also worked on a number of possible pathogens that could destroy crops or trees. And by the 1960s, the U.S. biowarfare program had begun to decline, with funding gradually decreasing, and they were growing in protests in the United States over the use of a defoliant in Vietnam. And I bet that's Agent Orange, and anger about a sheep kill incident in Utah, where the anthrax probably blew over the hills. That incident occurred in 1968, when 3,000 sheep were found dead in the Skull Valley area, which is adjacent to the Army's Dugway Proving Grounds. And although the findings were not conclusive, it was believed that nerve agents had somehow drifted out of Dugway during a test in aerial spraying. And they were doing a lot of research not only on bombs with pathogens, but also aerial spraying of pathogens. It's very, very frightening to me. In 1969, the Army announced that 23 U.S. soldiers and 1 U.S. civilian had been exposed to the sarin nerve agent on the Japanese island of Okinawa while cleaning sarin-filled bombs. Okay. The incident created international concerns and revealed that the Army had secretly positioned chemical munitions in Southeast Asia. That very same year, so it'd be 1969, President Nixon took action against biological and chemical weapons and reaffirmed that no first-use policy for chemical weapons and renounced the use of biological weapons and limited research, and this is the part I've quoted many times, to defensive measures only, and hence became our vaccine program. The Army had its beginnings during World War II. The Army fort that had its beginnings during World War II is now a sprawling campus of brick buildings outside Frederick, Maryland, located an hour's drive north of Washington, D.C. Government scientist Bruce E. Ivins worked at the heart of Fort Detrick inside the main buildings of the U.S. Army Medical Research Institute for Infectious Disease. It was there that he was one of the leading researchers on an anthrax vaccine. Oh, is that the one that they gave during the Gulf Wars that messed everybody up? The building houses a number of highly secure labs where anthrax spores would be used. And Ivan, who died on July 29th, 2008, helped investigate the deadly anthrax attacks in 2001. Oh, yeah. They didn't say how he died. He apparently had been notified that he was to be prosecuted, for the 5 deaths connected to the attack. Yeah, they didn't want him tweeting like a canary. So pretty interesting. So this is how it all ties back to the very beginnings. And I mean, it even goes back to the 40s. If you get into that biology of doom, they talk a lot about some of the early development of biological weapons and what some of the other countries were doing, such as England. And I didn't get to Japan yet, but I'm sure it's all in there. Now, speaking of Japan, we had also read a story about that Japanese Unit 731, which when they ended World War II, we went in there and wanted information on what type of work they had been doing, and we took a lot of the research that we uncovered and put that to work in some of the places like Fort Detrick and in Utah as well. So, speaking about Japan and a lot of the experimentation that they did on captured Chinese soldiers, the Pentagon is now worried, and this is March 1st, 2018, so this is just a month ago. We've talked about this before. Pentagon Lab worries about CRISPR bioattacks as China is seeking the genome lead. The contest for global supremacy in emerging technologies like hypersonic weapons and artificial intelligence may get the headlines, but the heart of the Pentagon's secretive DARPA research lab is concerned about the biological attacks that use this new gene editing technology. To be sure, cures for debilitating diseases could soon be in reach as companies like CRISPR Therapeutics, Intellia Therapeutics, and Adidas Medicine make huge advances with CRISPR, or the technology that allows biotech companies to edit, add, or remove faulty genes. But China is also investing heavily in human genome sequencing and is emerging as a leader in genome mapping with the largest database of DNA for its own population, says Stephen Walker, director of DARPA. They are taking it seriously, but the wealth of knowledge could be used for more nefarious purposes like a biological attack. We have asked leadership to really focus on and really think about how we do biosurveillance, using it to prevent biological attacks on the United States. Yeah, gotta love that spin. It's going to keep you safe. You know, maybe they shouldn't have democratized all this stuff. Hello, Clyde. Walker said that DARPA's medical lab is focused on synthetic biology and fighting infectious diseases, while neuroscientists, researchers are working to understand how gene editing works and develop some countermeasures to reverse a gene edit if it was ever released into the wild. And another medical project is DARPA's P3 program, which is an ambitious goal to create a transient immunity. Now, we've talked about this before. They put it in quotations, transient immunity. But this is the shape-shifting immune system stuff that we have covered for some devastating infectious diseases like Ebola or for a significant amount of the population within just 60 days. And the increased focus on China's biotechnology comes after the Pentagon released the 2018 National Defense Strategy in January with a shift in focus to, quote, great power competition versus insurgencies in the Middle East. So we are changing our focus from quote-unquote ISIS under every bed and in every closet to now CRISPR bio attacks. Now, we had covered a couple times, but we looked at it again on Wednesday, DARPA's Safe Genes program, and this was announced in July of 2017. They have given money to 7 institutions to develop a program that will be able to maybe potentially reverse and accidentally or on purpose gene edit to an organism. The efforts funded under the Safe Gene programs falls into 2 broad categories. One would be gene drives. That's something that DARPA and Billy Gates have invested heavily in. And genetic remediation technologies and in vivo, and that would be in a living organism, therapeutic applications of gene editors in mammals. So the teams that they have funded, one would be a team led by Amit Chaudhry of the Broad Institute in the Brigham and Women's Hospital Renal Division at Harvard Medical School in developing means to switch on and off genome editing in bacteria, mammals, and insects, including control of gene drives in mosquitoes for malaria. And the team seeks to build a general platform for the rapid and cost-effective identification of chemicals that will block contemporary and next generation genome editors. This is going to be a genome editor vaccine of some kind. Such chemicals could propel the development of therapeutic applications of genome editors by limiting off-target effects, which was always a problem, or protect against future biological threats. The team will also construct synthetic genome editors for precision genome editing. Well, good. So that's the Broad Institute and the Brigham and Women's Hospital of Harvard Medical School. Now, number 2, Harvard Medical School team, led by who? Oh, good old Georgie Porgy Church, seeks to develop systems to safeguard genomes by detecting, preventing, and ultimately reversing mutations that may arise from exposure to radiation. This work will involve creation of a novel computational and molecular tool set to enable the development of precise editors that can distinguish between highly similar genetic sequences. The team also plans to screen the effectiveness of natural and synthetic drugs to inhibit gene editing activity. Number 3, another Massachusetts institution. This would be Massachusetts General Hospital. Massachusetts General Hospital team, led by Dr. Keith Young, aims to develop novel, highly sensitive methods to control and measure on-target genome editing activity and to limit and measure off-target activity. Notice how off-targets are what they're focusing their attention on, since that was one of the Achilles heels of CRISPR technology. Where did I leave off here? And apply these methods to regulate the activity of different genetic editing functions. The Massachusetts General Hospital team hopes to enable orders of magnitude higher sensitivity than what is currently available with existing methods and make this process routine and scalable. The team will also develop novel strategies to achieve control over genome editors, including drug-related versions of these molecules as well. And the team will take advantage of contained facilities that simulate natural environments, so they're not going to be doing it in vivo, thank goodness, to study how drive systems perform in mosquitoes under conditions approximating the real world. That's disturbing. That's 2 stories where they are manipulating mosquitoes. Don't trust any little flying creature anymore, because they are rewriting biology and changing things like we never imagined. And there's a story that actually gets to that somewhere along the line here. The next institution is another one in Massachusetts, and that happens to be good old MIT. A team led by Dr. Kevin Esvelt, and we've run across him quite a few times, has been selected to pursue modular daisy drive platforms. I haven't heard of that one before, with the potential to safely, efficiently, and reversibly edit local subpopulations of organisms within a geographic region of interest. Oh, so targeted gene editing of local subpopulations probably identified by genetic markers. Daisy drive systems are self-exhausting because they sequentially lose genetic elements until the drive system stops spreading. And one of the proposed variants, which is natural selection, is anticipated to favor the edited or the original version, depending on which is the majority, and keeping genetic alterations confined to a specific region and potentially allowing targeted populations of organisms to be restored to pre-wild genetics. MIT plans to conduct the majority of its work in nematodes, well, thank goodness, which is a simple type of worm that reproduces rapidly, enabling high-throughput testing of different drive configurations and predictive models over multiple generations. The team then aims to adapt the system in the laboratory for up to 3 key mosquito species relevant to human and animal health, gradually improving performance in mosquitoes through an iterative cycle of model, test, and refine. Another institution is North Carolina State University, led by Dr. John Goodwin, and it aims to test a mammalian gene drive system in rodents. Now, this is kind of scary. The team's genetic technique targets population-specific genetic variants found only in invasive communities of animals, or maybe humans. And if successful, the work will expand on tools available to manage invasive species that threaten biodiversity and human food security, and that serve as potential reservoirs for infectious disease affecting native animals and human populations. So we are giving scientists the ability to wipe out entire populations of quote-unquote invasive species. How long is it going to be before we are considered invasive species? The team also plans to develop mathematical models of how drives would function in mice, and then performed testing in contained simulated environments to gauge robustness, spatial limitation, and, of course, the reversibility of the drives. Next is the University of California, Berkeley. Now, something that I'm finding here, too, is that the first 4 or 5 of these institutions that we had talked about happened to be in one of the stories that we read about for Fort Detrick as well. There was a whole group of universities that had received money for funding. And the University of California, Berkeley, with a team led by, who we know, Jennifer Doudna, she is one of the founders of CRISPR, will investigate the development of novel, safe gene editing tools for use as antiviral agents in animal models that target Zika and Ebola viruses. Now, we thought we forgot about those, didn't we? The team will also aim to identify anti-CRISPR proteins capable of inhibiting unwanted genome editing activity while developing novel strategies for delivery of genome editors and inhibitors. Here, just take a breath of this, Johnny. Get your CRISPR across that blood-brain barrier as we turn on some ultrasound. Boo! I mean, this is just crazy. Or let's eat your encapsulated lipid with the CRISPR tools inside. We read about that as well. All right, so the teams intend to refine our research over the course of the program, building initial mathematical models of gene editing systems, testing them in insects and animals to validate their hypotheses, and then feed these results back into the simulations to tune the parameters. Teams will also incorporate insights garnered from engagement with regulators and in some cases from local communities considering gene editing applications, and may run additional experiments on the population to collect data that addresses concerns and could inform future regulatory reviews. Given the potential gene editing systems to broadly impact national security, health, and environment, DARPA is committed to the highest level of transparency, of course they are, and engagement in its safe genes research. The program will work with independent experts to help DARPA and the teams think through legal, ethical, environmental, dual-use, double-edged sword, and responsible innovation issues. In a separate but related effort, DARPA previously co-funded the National Academy of Sciences, Engineering, and Medicine to report on gene drives to help initiate the development of a framework considering the implications of advances in gene editing and to make recommendations on a responsible way forward. So it's not as a weapon, right? Wink. I mean, this is crazy, but they're worried about China making a weapon, but we're not. No, this is all just in a defensive posture. And one aspect of safe genes that we are involved in from the very beginning, many of whom are already considering gene editing technologies as options for responding to different health and environmental challenges, but who have questions about how these solutions involving gene editors would actually work. DARPA sees their involvement in the Safe Genes program as invaluable for developing a model in which consideration of societal impact isn't an afterthought, but instead a foundation which forwards these science advances. Wah, 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 wah. I mean, what a spin. Yeah, I'm sure DARPA is looking at this only to keep you safe, people. It's just ridiculous. Okay, so everything that DARPA does is for the Pentagon, and everything the Pentagon does is in the name of warfare. So here's a story that came out of a publication April 19, 2018, called Laboratory Automation, Robots for Life Sciences. Now, we've discussed before a DARPA award that was given to the Broad Institute, I believe. And the Broad Institute was doing manufacturing of some sort of DNA or living cells. It was part of the 1,000 molecules program, I believe. But the Weiss Institute was also awarded $3.7 million from DARPA to develop what they are calling a genetic security system. And this story came out of the Weiss Institute October 20th, 2011. So 7 years ago, a new technology would provide bacterial background checks to enhance biological security and safety. The Weiss Institute for Biologically Inspired Engineering at Harvard University announced today, in October of 2011, that it was awarded a $3.7 million contract, including an option, which typically means they can extend it, from the Defense Advanced Research Project Agency, more affectionately known as DARPA, to develop a genetic security system that would track an organism's history. The proposed DNA-based memory device would sit inside of a bacterium and create a permanent record of the historical experiences in much the same way as the track changes feature of a word processing program saves successful edits in an electronic document. Such a bacterial background check would be analogous to a biological forensics tool. Ding-a-ling. They'll be able to tell everything you touched, everything you ate, everything your cells did. And this could be considered similar to a fingerprint analysis, DNA testing, or blood typing. This DARPA project will be led by Weiss Institute core faculty member Pamela Silver, who is a professor of systems biology at Harvard Medical School and the first director of Harvard's program in systems biology. She is also a co-principal investigators will also be James Collin, who is a Weiss Institute core faculty member, as well as a professor of biomedical engineering at Boston University, and Jason Kelly, a founder and principal scientist at the Boston-based startup called Ginkgo Bioworks. We've run across them a few times in stories, and I think one of these other stories mentions them as well. The project team is charged with overseeing development of a bacterial memory system that is actively reporting on and tracking the history and the status of an organism, providing information on specific experiences such as exposure to an antibiotic or maybe even a pathogen. This type of tracking system could protect commodity biomanufacturing by tracking the theft of proprietary bacterial strains that have been metabolically engineered to produce high-valued products such as biofuels or chemicals. It could also enhance the security of bacteria that are being studied in the laboratory and discourage the misuse of dangerous biological pathogens. The device would need to be robust enough to function in the field while also maintaining accurate historical records in the face of a wide range of environmental stresses, including the death of the bacterium that is charged with tracking. This would be one of the very first DNA-based memory systems to accurately track bacteria, and it represents just the kind of challenging and potentially game-changing work that we do here at the Weiss Institute of Biologically Inspired Engineering. We are happy to collaborate with DARPA, of course you are, in creating a new way to help ensure that bacterial strains created for science and industry are not misused in ways that could potentially harm people or endanger access to important products. Now, what happens when some of these tracking bacteria get into your probiotic and end up in your gut? We read about that before. Billy Gates had invested in some of that technology through MIT, and it was called... let's think. Living Therapeutics, I believe. And they wanted to put these little modified bacteria into your gut to be able to bag, tag, and track and cure you and make medicines inside of you. So, okay, September 29th, 2015, DARPA gives MIT lab $32 million to program living cells, pushing synthetic biology to the next level. Just yesterday, the Broad Institute, which is a synthetic biology laboratory at MIT, announced a brand new contract with DARPA. The lab will receive $32 million for engineering cells to find better treatments for disease, make new biofuels, or create fabrics woven with life. Living cells are the ultimate engineering substrate, and they're the most difficult thing out there to be able to control. Imagine being able to engineer a living cell that can navigate the human body, identify disease, and then correct that disease. That will require that the cell is able to sense where it is in the body and be able to detect it and then deliver a therapeutic. And that's something that biology, we know it can do. We just don't know how to harness that as part of a medicine. The Foundry is one of the many labs working to manipulate DNA of bacteria and other types of cells to make certain molecules, and researchers have created cells that can make wood or create seashells, for example, and the work has mostly been limited to simple organic molecules, and the progress has been slow since it takes a while for DNA to be put into cells and then for those cells to mature. And with the DARPA contract, this foundry will be able to join forces with other academics working on synthetic biology and computer science, as well as companies in various industries, including chemical, pharmaceutical, food, energy, agriculture, and biotechnology, says the press release. What specifically DARPA hopes to gain from works done at the foundry is vague. Of course it is. 2 years ago, DARPA granted the lab $7 million in seed money to add increasingly complex and numerous genes to engineered organisms. The lab had already created hundreds of man-made nucleotides and had even found less resource-intensive ways to make fertilizer. Well, that's good. And while the press release states that the grant will enable the foundry to, quote, offer critical new products in human health, agriculture, and chemistry, and serve as a mechanism for tackling some of the biggest problems of the world, and there's the spin, over the coming decades, it is not specific about what projects it will take on. Since DARPA is constantly out looking for new ways to hack human biology and create new materials for soldiers and artillery, it might not be too much of a stretch to speculate that medicine and biomaterials will be near the top of the lab's laundry list. Yeah, which reminds me, there's one other story talking about this investing $32 million into DNA manufacturing. DARPA investing $32 million into DNA manufacturing. Our super soldiers on the way. We've talked about the super soldier program before. September 26, 2015. This is out of the techtimes.com. The Defense Advanced Research Project Agency, more affectionately known as DARPA, has awarded $5 $32 million contracts. Oh, a 5-year $32 million contract to the Foundry as part of the agency's program known as the Living Foundries 1000 Molecules. The said program aims to establish facilities where scientists can rapidly engineer cells as a way of producing chemicals and materials which are otherwise not produced naturally. Society relies on many products from the natural world that have intricate materials and chemical structures, and from chemicals such as antibiotics to materials like wood, says Christopher Voigt, a biological engineering professor at MIT. We've limited our ability to program living cells to redesign these products, and I want to change the scale of genetic engineering to access anything that biology can do. Diddle-a-ling. 2 years ago, the Foundry started to work on a project that focused on the assembly of massive genetic systems wherein a number of genes are involved, and the facility is formed as a result of a joint venture by Broad Technology Labs and MIT's Synthetic Biology Center, where Voigt is a co-director. It earned a total of $7 million in seed funding from DARPA. And in order to meet the goal with needed efficiency and technological innovation, the foundry has sought to work in collaboration with partners from academic and industry sectors. See, that's how the tentacles happen. Give them lots of money and they get everybody to jump into the goopy soup. One of the academic partners is also Associated Professor Michael Fischbach of the University of California in San Francisco. This foundry has made it possible to do something that used to be a figment of my imagination, says Fischbach. Another collaboration was made with a consortium member from the Dutch multinational company called DSM on biotech and materials. And the joint effort resulted in the design and delivery of synthetic DNA that measured up to 6 million nucleotides, almost equal to 2 full bacterial genomes. The foundry is at the forefront of synthetic biology development, and it should be remembered that in 2013, DARPA also launched a solicitation effort for the project called Advanced Tools for Mammalian Genome Engineering. That would be you and me. And more than just being focused on the engineering of any mammal's genome, the project was specifically launched for the bioengineering of humans. The successful development of technologies for rapid introduction of large DNA vectors into human cell lines will enable the ability, this is a quotation, to engineer much more complex functionalities into human cell lines than are currently possible, states the project's proposal page. In other words, DARPA for several years now has been trying to defy what nature can offer to find ways to solve the limitations of the current gene transfer technologies and how to improve existing approaches in order for science to finally displace natural life. How crazy is that? Oh, so going back to biowarfare and the quote-unquote ending of our biological warfare program. No, it just kind of mutated a little into synthetic biological engineering. But some of the same partners are still involved, and the Department of Defense. All right, so now let's get to some mad science gone mad, down long, dark hallways, and some of the crazy things that they are doing. We have talked about mini brain balls before, and there have been quite a number of stories, and they've brewed up some of the batches of the creepiest brain balls yet, as they let them grow for longer periods of time in petri dishes, and then injected them into the brains of little mousies, where they then integrated with the circulatory and the neural nerve system all on their own without us doing anything. Well, let's look at some ethics here. You know, this is something that I like to look at every once in a while. And I mean, really, this is the whole premise of the show. Their ethics are completely out of window. And it is mad science gone mad. And everybody is racing for the cure to benefit you as they are utilizing biology as their guinea pig. The ethics of experimenting with human brain tissue, and this is out of Nature, which is one of the world's foremost science publications. Difficult questions will be raised as models of the human brain get closer to replicating its function. This was quite recent. I think it was April 25th, 2018. So this was just a couple days ago. If researchers could create brain tissue in the laboratory that might appear to have conscious experiences or subjective phenomenal states, would that tissue deserve any of the protections routinely given to human or animal research subjects? Well, this question might seem a little outlandish, but certainly today's experimental models are far from having such capabilities, but they're getting there. But various models are now being developed to better understand the human brain, including miniaturized, simplified versions of brain tissue grown in a dish made from stem cells called brain organoids. And advances keep being made. We even read in one of those extended little mini brain balls that it had started to develop olfactory tissue that would allow it to smell, giving it one of the many senses. What's going to happen when it grows little googly eyes? And it can start interpreting its position in space. These models could provide a much more accurate representation of normal and abnormal human brain function, and the development of many animal models can, although animal models will remain useful for many goals, these are even better. And in fact, the promise of brain surrogates is such that abandoning them seems itself unethical, given the vast amount of human suffering caused by neurological and psychiatric disorders that we might be able to fix with a little mini brain ball, given that most therapies for these diseases developed in animal models fail to work in humans. Yet the closer the proxy gets to the functional human brain, the more ethically problematic it becomes. And there is now a need for clear guidelines for research, because it's just all mad science right now, albeit ones that can be adapted for new discoveries. This is the conclusion of many neuroscientists, stem cell biologists, ethicists, and philosophers, ourselves included, who gathered in the past year to explore the ethical dilemma raised by brain organoids, i.e. little mini brain balls, and related neuroscience tools. The workshop was held in May 2017 at the Duke Initiative for Science and the Society at Duke University in Durham, North Carolina, with limited support from the U.S. National Institute of Health Brain Initiative. Dilling, that brain initiative that Obama inked. And a similar U.S. meeting was held last month on related topics. Here we lay out some of the issues that we think researchers, funders, review boards, and the public should discuss as a first step to guiding research on brain surrogates. So they're not called mini brain balls anymore. They are called brain surrogates, maybe even in preparation for one day being able to upload you and back up your memories into another vessel that could outlast your physical form. And that's Kurzweil's wet dream. All right, 3 classes of brain surrogate offer researchers ways to investigate how living human brains work without the need for potentially risky, if not ethically impossible, procedures in people. Brain organoids can produce as much as other 3D multicellular structures resembling eyes, gut, liver, kidney, and other human tissues have been built, and by adding appropriate signaling factors, aggregates of pluripotent stem cells, ding ding, those iPS stem cells, which they can grow even from your skin. And I saw a brand new story, and I don't know if I pulled this one up. If I didn't, I'll have to go find it. But I found a story just from the other day that talked about them being able to use stem cells even from elderly people to grow brand new cells. So that's a huge deal. By adding appropriate signaling factors, the aggregates of pluripotent stem cells can differentiate and self-organize into structures that resemble certain regions of the human brain. Investigators use different approaches, and they might coax pluripotent stem cells to turn into specific populations of neural cells, but such are specific to each particular brain region. And they even found, in one of those other stories we covered, the ones that they let grow for a long time, that the cells started to differentiate just like those in a fetal brain. So that was pretty scary as well, that they would be able to maybe even grow a whole human brain. And if they put it into a mousey and maybe even into a primate, though they didn't discuss that, but why wouldn't they, in the future, and it grows its own vasculature and nerve connections, who's to say that they're not going to grow replacement human brains with your neurological imprint? I don't know. This is just nutty stuff. Or they can allow the pure pluripotent stem cells to differentiate on their own, like I said, in which case both neural cells and other cell types might be generated. Brain organoids resembling particular brain regions can even be combined into what they're calling a quote-unquote brain assembleoid, like Legos, to enable researchers to study the formation of neural circuits and the cellular interactions between the different regions. Compared with 2D sheets of neural cells in a dish, these 3D structures last a lot longer, and they've found so far for around 2 years, and can consist of more types of cells. They also mimic key features for developing brains. For instance, in later stages of fetal development, the cerebral cortex switches from generating neurons to creating glial cells, which are various other types of cells in the brain that nourish, surround, and protect neurons. This process can be captured in a brain organoid, allowing investigators to gain insights that would be experimentally and ethically extremely challenging, if not ethically unacceptable, to obtain from developing brains. And already researchers have deployed brain organoids to investigate neurodevelopmental alterations in people with autism and schizophrenia, and to study the unusually small brain size, microencephaly, as seen in some babies infected with the Zika virus. Brain organoids have limitations. Of course, they were going to find a way to justify studying for Zika. Brain organoids have limitations, though. They lack certain cell types, such as the microglia and cells that form blood vessels, but that's where the implantation comes in. And today, the largest organoids are about 4 millimeters in diameter and contain only about 2 to 3 million cells. An adult human brain measures roughly 1,350 cubic centimeters and is made up of 86 billion neurons and a similar number of non-neuronal cells. Moreover, so far, the brain organoids have received sensory input only in primitive forms, and the connections from other brain regions are limited. Given such constraints, the possibility of an organoid becoming conscious to some degree or acquiring other higher-order properties, such as the ability to feel distress, oh, that's so horrible, seems highly remote. But organoids are becoming increasingly complex. Indeed, one of the recorded patents of neural activity from an organoid, one of us, PA, so I guess that's a patent, that's just whoever the initials are, recorded neural activity from an organoid after shining light on a region where the cells of the retina had formed within the brain. Oh, so I forgot about that one too. So not just olfactory neurons, but also retinal neurons. And this illustrated that an external stimuli can result in an organoid response. Another type of model involves slices of brain tissue that have been removed from individuals during a surgical procedure to, for example, to treat seizures. Oh my God, don't ever get that done. For more than a century, researchers have studied brain cells and tissues extracted from patients undergoing surgery. I thought they were doing that up in the, what's his name? The guy that was part of Microsoft, the Allen Brain Institute. We read about that. They were actually studying slices of brain tissue from surgery patients. For more than a century, researchers have studied brain cell and tissue extracted from patients undergoing surgery or from people who have died, but technological advances, including in imaging and in techniques used to preserve the functional properties of brain tissues in the lab, could make this approach considerably more powerful. When tissues from the neocortex or the hippocampus region is removed to treat a pathology such as epilepsy or cancer, the piece removed is typically the size of a sugar cube, although can sometimes be much bigger. Don't sign up for that. That piece is then generally cut into slices, and the functional properties of which can be preserved for weeks. Using these slices of brain, researchers can measure the synaptic and other properties in the neurons in the intact brain circuits and then map the 3D morphology of the circuits and then extract and analyze the cellular RNA to probe the gene expression. They can also manipulate the firing of specific neurons using optogenetics, ding ding, which could then enable them to analyze in more detail the functional properties of the human brain circuits. And then they explain what optogenetics is, using light to turn things on and off. Currently, ex vivo, and that would be stuff that is outside of a living organism, brain tissue does not have sensory inputs, and with outbound connections severed, the isolated tissues cannot communicate with other brain regions or generate motor outputs. Thus, the possibility of consciousness or higher order perceptive properties seems extremely remote. But then the third experimental brain model is, chimeras. We have talked about these before. These are those brain chimeras. Third class of experimental brain models involves the transplantation of human cells derived in vitro from pluripotent stem cells into the brains of animals such as rodents. And we have read a couple different studies on that. This can be done while the animal is a fetus and is developing or after the animal is born. And such chimeras are generated to provide a more physiologically natural environment in which the human cells can mature. And in the latest experiment I read about this, they replaced the top of the little mousy skull with a clear, like, plexiglass panel so they could actually see the human cells growing in vivo and growing vasculature inside of the mouse brain. I mean, it's just, I can't even get over how disturbing this is to me. And you have to think, this research is going on. It's all in the name of curing you or keeping you safe. This isn't going to be isolated to the laboratory forever. Neuroscientists have transplanted human glial cells into mice, for instance, and found that the animals perform better in certain tasks that involve learning. Of course they were. Researchers have also injected human stem cells into early-stage pig embryos and then transferred the embryos into surrogate sows, where they were allowed to develop until the first trimester. And these were the pigs, I think, that they were going to use to grow human organs that Georgie Porgy Church was involved in. Xenotransplantation or something like that. More than 150 of these embryos developed into chimeras. In these embryos, 1 in every 10,000 cells is a precursor of hearts, and the livers were human. Oh, God. So these were human livers. In principle, chimeras could help researchers to better understand human illnesses and treat the effects of drugs. Labs have developed human mouse chimeras to shed light on Parkinson's disease, for example, and some groups have even successfully transplanted human brain organoids into rodents where they have become supported by blood vessels, ding ding, and that would be called vascularized. The provision of a blood supply is an essential step in enabling organoids to grow larger than the currently achievable size. But the size of a rodent model restricts the degree to which human brain organoids can grow within them. And that's why I say I bet the monkey is next. Currently, the research on human tissue occurs outside of a living person. And only the process of obtaining, storing, and sharing, and then identifying the tissue falls under the regulations and guidelines that limit what interventions can be conducted on people. As brain surrogates become larger and more sophisticated, the possibility of them having capabilities akin to human sentience might become less remote, and such capabilities could include being able to feel pleasure, pain, or even distress, and being able to store and retrieve memories, or perhaps even having some perception of agency or self-awareness. Now, this is all out of Nature magazine. So we're getting pretty far into the show here, and I have a bunch of other stories I want to hit. So now let's jump to Out of Nature, and we're going to go to theatlantic.com, April 25th, 2018. This was 2 days ago. The title of the story is What is Wrong with Growing Blobs of Brain Tissue? Well, very good question there. These increasingly complex organoids aren't conscious, but we might not know when they cross that line. Last week, Rusty Gage and colleagues at the Salk Institute announced that they had successfully transplanted lab-grown blobs of human brain into mice. And I thought it was longer ago than a week, but maybe they just finally published it publicly a week ago, and we had read about the research. Gage's team grew the blobs, known as brain organoids, from human stem cells, and once surgically implanted into rodent brains, the organoids continued growing and their neurons formed connections with those of the surrounding brains. It was the very first time that such a transplant had worked. Until now, organoids had only been grown in dishes. To be clear, Gage's mice weren't running around with human brains, nor did they have human minds trapped inside their heads, called little mousy chimeras. The biggest brain organoids are lentil-sized and contain 2 to 3 million cells, and a human brain is 20,000 times bigger and contains around 172 billion cells. Even the biggest ones don't have a full set of cells needed for a working brain. So on their own, the neurons don't form networks like those in our head. They don't sense, learn, or make memories, yet they are emphatically not brains in jars. They are not little mini brains either, so I stand corrected. In the same way that a leaf is not a mini plant and a doorknob is not a mini building. That's a good illustration. They do, however, capture some of the architectural features of parts of the brain, and that's why they are useful. Scientists can use them to understand how brains develop and how they differ in disorders. For example, one group of researchers made organoids with a genetic mutation that is linked to microencephaly. Those organoids were also smaller than usual, and the team could find out why. Another group, led by Hung Zhang at the University of Pennsylvania, used organoids to confirm that the Zika virus can affect fetal brains and cause microencephaly, and to pinpoint which cells it infects. There are also many issues why psychiatric disorders have been hard to crack, but one that we don't have access to is a functioning human brain. Says neuroscientist at Stanford University, imaging technologies are still relatively crude, but always getting better, and animal brains are similar to ours, but they grow differently. Organoids can also be limited in their own ways, allowing us to capture aspects of human brain development that were previously not accessible. So, growing human brains. Now, there was another one out of Nature, the boom in mini stomachs, mini brains, mini breasts, mini kidneys, and more. Biologists are building banks of organoids and learning a lot about human development along the way. It was an otherwise normal day in November, and this was from July 29th, 2015. So this was 3 years ago, when they had accidentally grown a brain at the University of Lancaster. And for weeks, they had been trying to get human embryonic stem cells to form neural rosettes, which are clusters of cells that become neurons. But for some reason, their cells refused to stick to the bottom of the culture plate. Instead, they floated. So they really didn't know what they were. Well, they were not the first to grow brains in a dish. In 2008, researchers in Japan reported that they had prompted embryonic stem cells from mice and humans to form layered balls reminiscent of the cerebral cortex. And since then, efforts of growing stem cells into rudimentary organs have taken off, and using carefully timed chemical cues, they can make them into anything they want for three-dimensional structures that resemble tissues from the eye, gut, liver, kidney, pancreas, prostate, lung, stomach, and breast. All right, guys, give it a